Okay, moving along with the next company. First, I just want to emphasize and remind everybody that listening in that you can post your potential questions through the chat forum. So use that possibility to ask questions to the company. So our next company to present is FluoGuide, and here to present is Morten Albrechtsen, its CEO. Go ahead, Morten.
Thank you very much for the invitation and welcome. I'm really pleased that I can present FluoGuide now. We have had an extraordinary interesting equity story the first 2 years on our exchange since 2019, and we have every reason to believe it will continue. So what we do is that the FluoGuide, we maximize the surgical outcome. And first that you should make your own assessment. So we start out in oncology surgery. And the reason for that is that 15 million people every year have cancer, and 80% of them need surgery. And half of this cancer is recur after surgery. So that means every every second patient will have cancer locally after surgery. And this is not because the surgeon are bad or poor in any way, just because it's very hard to see where the cancers stop and where the normal tissues start. And our solution to it is simple. We have a new part targeted the illumination of the cancer, and that way we can help almost 80% of the patient undergoing cancer surgery. So our first product is under clinical development. It's been demonstrated to be well tolerated, and it demonstrated that it lights up cancer. So we see very much now that we have a product. Now it's a question of getting it out to as many patients as possible. We have to prove the maximum benefit for the patient, but also the price. And that's what we are working on the next couple of years. We have a good platform. We have an IPR protection on a field, UPAR guided surgery. We have a good cash position. We are on the first north here in Sweden with a market cap about 1 billion SEK. And then we have a team that is quite functional and actually cover the relevant fields. So first, our procedure. It's a very simple procedure. Before surgery, the patient have our product injected into a blood vessel, and it will circulate in the body, and it will bind to the cell that express UPAR. I'll come back to UPAR in a second, but it's a very cancer-specific target. So it will bind to the cancer, and it will, when it's removed from the background, it will just remain on the cancer and light up the cancer. Then during surgery, the surgeon can switch on the fluorescent light, and they're then able to see precisely where the cancer is and can do a precise surgery. I think one of the three things that's very important with FluoGuide is that everyone really likes us. I mean, obviously the patient likes us. They get a higher likelihood of being a complete surgery. The hospital can potentially save procedures or more aggressive treatment following surgery that has been not completed. The surgeon obviously also like us because they don't like to go back and tell a patient that the surgeon was not done fully, and that they have to undergo some more aggressive treatment or another surgery. So they actually like to go back and tell the patient that all the cancer have been removed. So they like being helped in this sense. And last but not least, equipment manufacturers also likes us because we're able to get more features out of. That means that we have a possibility to get more benefit out of their technical features on their equipment. So there's a lot of equipment that can do much more, and we are actually able to put that into play to the benefit of the patient and to the equipment manufacturers. So what we see is a very strong interest from the equipment manufacturers in seeing us succeed because that will help them as well. So UPAR is a target that is being worked on for a long time. It's an enzyme system that is sitting on the forefront of the cells, bound to the surface of the cell. And what it does is that it helps the cancer to break down the normal tissue so it can spread. And that means that it will particularly be expressed on the forefront of the cancer, and they will be particularly expressed on cancer that is malignant. And that means that the more malignant the cancer is, the more UPAR is expressed. And the more is expressed on the forefront of the cancer. And that's really where the surgeon needs to see it, because that's where it's difficult to delineate the cancer from the normal tissue. So there's been a large body of scientific literature on UPAR. It's been used as drug target, it's been used for diagnostic purposes, and now we use it to guide surgery. And we have done a couple of preclinical studies to demonstrate that it works. And one example of that is this study where we took a human surgeon, we took a human cancer and human equipment, and we implanted the human cancer into a mice, and we asked the surgeon, we had 8 mice in this study, we asked the surgeon to remove all cancer under white light. And in the 4th of the animals, he removed more cancer. I mean, he could see the cancer and he removed it. But from the 4 more of the animals did first have cancer removed after the fluorescent light was switched on. And had it been patient, they would have been sent home and were told that the surgery was done successfully, and that then would have come back at a later stage, either because the cancer have regrown and they had it was to be found in a screening, or maybe they can ultimately be dying of the cancer that has spread. But that's really what we can do in the clinical studies as well, and the clinical practice, that we can help that additional number of patients seeing with the surgeon that they can see the cancer and remove everything. Another thing we did here was that we use standard equipment. That's one thing that is very important for us, is that we do not want to expect that the whole equipment package out with the surgical theaters have to be changed or modified. We would like to have a plug-and-play situation where we can use a standard equipment. We don't want to be only used for the most sophisticated equipment. We want to have a product that is good enough for the, I would say, poorest equipment that's out there. So we really have a situation where we don't have to go out selling equipment, but we can go out selling our procedure, and we have an easy adaptation of our product. So our initial plans will target more than 5 billion euro, and that's a combination. I mean, this is just some of the key cancers and the incidence. And you can see there's some cancers that have a not that frequent prevalence, but they have an orphan drug designation potential. And then there are other cancer, breast, lung, and colorectal cancer, that have a very high incidence, but that will of course not be orphan drug designation cancers. And we have decided on a mix of indication between those, so we both get an indication where we can get fairly quickly and safely to the market, and then we can expand into the more prevalent indications. So the first indication we have selected is high-grade glioma, or aggressive brain cancer. Glioblastoma is also called. It's several names. And a couple of reasons for that is that brain is a very difficult organ to make the surgeon in, because you need to take out all cancer on the one side, but you need also to be very careful that you do not take too much, because that will disable the patient. So if you, for instance, say you remove the center of speech, the patient cannot speak afterwards, or it could be movements that are being impaired. So precision really matters in the brain. Another interesting thing with this indication is that the neurosurgeon are well trained for using equipment. Microscope has been used for a long time in a neurosurgeon, because it's really very detailed and very precision surgery that is important. And our product will fit into the setting of the microscope. So that's another reason for us choosing it. Then the second thing that is very important with FluoGuide is that we have a very short way to the market, and that is because we have a short and simple, small clinical trials needed to support approval. Basically, we need 2 studies that is done almost similar, in a sense, just a number of patients. A patient would be included into the study in 48 hours, so we include them, inject our product, and then we look at the surgery, and then they are excluded. And that's because the endpoint that is needed from a regulatory purposes is what's called positive predictive value. And what that means is that when the neurosurgeon sees some tissue that lights up, they take a biopsy, is sent to the pathologist, and the pathologist validate that it in fact is cancer. And if that's the case, then it's a positive predictive value, meaning that when something lights up with our product, it will be cancer, and demonstrated that it's cancer. So we need 48 hours per patient. And I said we need 2 studies. The first study, we have to demonstrate safety. We have to select the dose, and we get a proof of concept of this study, and also we get an idea of the magnitude of benefit we can expect from our product. Then the second study is actually designed very similar to, but just with more patient. There we demonstrate safety in a larger population, and then also efficacy, meaning this positive predictive value. We would expect or anticipate that we need approximately 150 patients for a pivotal study for glioblastoma. Our first study that is ongoing right now is phased into two. It's a part 1 and 2. The first part of it is a safety study, but what we also is in patients. So what we also gain there is information of our product if it lights up the cancer. And what we see is actually that it's very precisely light up cancer. I mean, that is it's a nature of our clinical studies that we cannot blind it because no surgeon can see the light. And if she can see the light, I mean, she can see if it's cancer or not. So we know it actually lights up cancer. We also know that it's well tolerated, and what we have promised is that we will report the safety now here in Q3, and that means we are close to an end of this first phase in a sense that we have a dose that we know that will be optimal, and then we can go into the second part of the study, really, which is a mini phase 3 study designed exactly similar, and really used to do the power calculation for the phase 3 study. We have had this 5 cohorts, 21 patients included. Our product have been well tolerated, as mentioned. This is also what we expected from the preclinical package, because what we have seen there is that we have pushed up to, we have to see either toxicity or lack of feasibility. If we cannot get more product into the animals, we will be limited by that, and we have been limited by feasibility, not toxicity. And we see a dose that is much higher than what we need in the clinical practice, 100 times higher. So we have a huge room up for the limit where we can dose in patient. It's not the toxicity limit, but the limit for the patients. So we really anticipate that it would be well tolerated, and that's also what we have seen. So the pipeline of the company now, we have the first product. It's the lead indication is this aggressive brain cancer, and we move it into the pivotal part. I mean the phase 2 part in Q1, and we also will move it into phase 3. We prepare the phase 3 next year, and then end of next year we move into phase 3 with the study, and that really convert the company in for being a late stage company. You may have recognized that we have a Swedish approval now for adding the Swedish site in our first study, and that's we do because that's required by pivotal part that we have more than one country, more than one center, as we have lined up with the Swedish center as well and got approval for getting that started. Then we will initiate a prevalent indication. We did a directed issue here before summer, and part of that proceed will be used for this study. We haven't announced yet what it will be, but it will be either lung or breast cancer, and we will announce it very shortly which of the two. So we are in deep development or preparing for this protocol and submission. And as soon as we get the safety report for our first study, we kind of have a license to hunt, and we can actually submit in all the indication we want that potentially is relevant. But what we want to drive ourselves and the company is one indication that get us quickly to the market. That's a aggressive brain cancer, and then one indication that go out for a prevalent indication that will benefit many patients, and that is, as mentioned, lung or breast. Then we have the second product coming up that was licensed in the middle of last year, and also part of the proceed we got here for before the summer will be used to initiate the development, and that has been started now. The development we see some longer timelines for toxicity studies and so on due to the corona. So we will not yet put on except milestones on it. We are getting the vendor deal and get a lock-up agreement with them before we announce exact development timelines. But we have started the whole process, both getting it produced and get the toxicity testing. And the reason for us why we want to have two products is that it gives us much more flexibility in commercialization. We can do price differentiation. It also gives us more flexibility in the partnering, in the sense that we can sell off an entire molecule, for instance, for an indication, and it's quite nice that we have multiple indications we can work with, and it would be nice to have this flexibility as we go on. Then there's also listed this photothermal therapy. That was something that we just had an article that was published here recently. And really what that does is that when we light on our fluorophore for FD1, when we light on it, it will be with very high power intensity light. We will see that it will heat up. And as it is very specifically bound to the cancer, it will kill the cells that it's bound to, and that means the cancer cell, but it will not kill the cells around it, meaning the normal tissue. And that's quite neat functionality we had with this FD1. And an example of that is, for instance, on the brain, where you in 30% of the cases will have a neurosurgeon that will not remove all cancer, simply because the cancer have invaded into a critical function of the brain. It could be the center of speech, and they just leave the cancer behind, because if they removed it, the patient will be disabled and in fact will get a less good treatment post-surgery. So the best for the patient actually is leaving cancer behind. And this is a well-known problem for the neurosurgeon. And our photothermal therapy would be a great tool for them, because that could actually make us sweep this area, or the surgeon to sweep this area, and allow them to actually remove the cancer cell without destroying the critical or sensitive function in the brain. This is something we expect a lot from, and we are currently doing all the planning and will move into development of this feature of our first product sometimes next year. In the field of competitors, there are competitors out there, and we actually see them both as benchmark cases, and we also see them as helping and supporting each other in developing a novel field. We see two products out there that is approved today. One of them is Hexvix from Photocure for bladder cancer. Another one is SBI, is a Japanese group, for Gliolan, is for brain cancer. And that's also one of the reasons why we have chosen brain cancer, because we have a reference product that we can lean up against, and we can get a more generic-like approval that will help us in decreasing the process risk in the regulatory process. But one of the problems with those products, this first generation product, and they're very good as such, but one of the problems with it is that the fluorescent molecule is created by a metabolite of the drug that is injected, and that will make them forming a kind of cloud around the cancer, so they're not specific to the cancer. Another issue is that they are single indication. You cannot spread them out for other indications. And the last thing, which really is important, is that they're based on visible light, so you can only see them on the surface of the tissue. You cannot see them into the tissue. So the rest of the product shown on a slide is based on near-infrared light, and that you can see 1 to 2 centimeters into the tissue. And imagine, for instance, on the brain, where you really want to see into the brain, see into the tissue, it's a quite important feature. If you have a product where you only can see it on the surface, you have a two-dimensional image, whereas you see it into the tissue, you can actually imagine the cancer underneath into the tissue, and that's a very strong different thing that is recognized as an important thing for our product. But of those companies that's listed here, 3 of them are equipment dependent, and that means that you have a special equipment needed for utilizing the product. It's one of the things we would like to avoid, to have a more flexible and faster penetration into the market. And then it leaves 2 product company behind that I think is important. One is SurgeryMap, and the other one is OnTarget. And they have, one of them is in colorectal cancer, the other one OnTarget is in ovarian cancer and lung cancer. And they have just submitted their application for ovarian cancer, and that could get a feedback from that in the next 6 to 12 months. I think, and for our product with the UPAR target, that is a very cancer-specific target on the one hand, and on the other hand, it's cancer type unspecific. So we can go in on the glioblastoma, high-grade glioma, in an niche indication, and then we can spread it out to the more prevalent indications. And that's really what we think is quite neat with the UPAR target we use for guiding surgery. I think that it will not be, in the end of the day, FluoGuide will not dominate the whole field. I think that we have to share the market between us, but I think that the fact that there are multiple companies in the field will help developing the field and maturing it, and I would expect that will happen a lot of this in this field the next 5 to 10 years. The next thing, third thing that is interesting for FluoGuide is that we have some very interesting trends, mega trends in the surgical field. in the old days, the surgeon used a knife and the hands and the eyes, but now they've been helped by equipment coming into the field. That's microscopes from the neurosurgeons. It's scopes from for instance, for lung physicians or surgeons, and then also the robotic surgery is an important tool that have huge success in the last decades. And they all need one thing really to get their features, enable their features, and that is something that can guide them. And all the fluorescent guided markers, not only FluoGuide, but also the other companies I mentioned on the other slide, the previous slide, could actually help them guiding and steering the equipment. And that is probably the reason why we see a huge interest for those kind of companies to see us succeed, because if we succeed, they actually also will succeed themselves. But as I mentioned, the glioblastoma, high-grade glioma, was one of the indications we focus in on, because the neurosurgeons are using the microscope, and that actually deeply penetrated into the neurosurgeons field. And that is already set up for seeing our product, so we don't have to go in and redo it or sell extra lasers or extra filters or anything like that. We can just really plug and play. So that's why we start out there. But that is a very interesting thing. We see this surgical field is having a booming field. Intuitive surgery is an example of a US company. Surgical science is an example of a Swedish company that have a lot of success in this field, and we expect also that we will be quite important driver for FluoGuide. The team behind, we are keeping the quite a small team to start with. We can of course not keep it small for a long time. We have to scale up at some point in time, and the time where we will really significantly scale it up would be at the time we start going into phase 3 with our first product. I mean, there's a lot of quality assurance and things like that we should take care of, and that will need some people. But for now, we keep it small, and we have picked people that are very highly skilled in each of their respectively areas. So on everything starts here with Andreas Kjær. He's board certified nuclear medicine, and he's working with a group about 50 people in Copenhagen University, working on UPAR, imaging, cancer, and diagnostics. And we are spin out from his department. He's also working on a hospital, and that's where we're doing our first studies right now. So he's really a very high-profiled scientific person. Then the next one in the line is Grete, and she is very strong on the CMC. We are under the drug regulation, the pharmaceutical regulation, and Grete is taking care of all the production and is doing that very, very well. She I think was employee number 4 in Ascendis Pharma, a US company that just had a product approved a few days ago, I think it was, and she's done all the CMC for that, and I think she is extremely skilled in the CMC and really doing a very high quality development, which is important because we don't want to have hiccups in the CMC, and that is often a source of hiccups. The next one in line is Dorte, taking care of the clinical operation, together with Andreas doing the more design of the studies. Then Henrik is a seasoned CFO with a lot of experience in life science companies. For the first one he was participating in was NoSearch, and then a lot of companies since that. Myself is MD and have a commercial background. I've been in sales my time, and I'm working as an entrepreneur since 2000. And the last company I did was an oncology company, it was a medical device, taking a product from the bench to the market, and now it's FluoGuide's turn. We are also, as mentioned, listed on a First North here in Sweden, and beginning of the year we moved from Spotlight. We had been remunerated or was awarded as the best IPO in 19, and that was we appreciated that a lot, of course, but really would like to continuing a strong and a proper development as we have done so far. We had an institutional 15 institutional investors that came on board before the summer, and we raised 75 million Swedish crown, and that's quite important for us when we mature, that we have more support, and was Swedish, Norwegian, Danish and German investors that came on board. We have an EU grant from the European Union, 2.5 million euro, halfly paid out. And then we have two institutions that were supporting us from the very beginning, and one is Link here from Sweden, and then we have a mid-sized bank in Denmark that is supporting us. And we have a quite large number of shareholders. So we think FluoGuide is a very interesting investment opportunity, and we think that we can continue to deliver quite exciting milestones. So we have in Q3, we have the safety, we have preparation of a prevalent indication being quite important for our value as we go on. We will have started the development of this FD2, the second product, and next year we have efficacy coming out in steps of more validity, and also that we prepare the phase 3 trial for FD1, which will be major transforming the company into a late-stage company. So thank you.
Great. Thank you, Morten. I think there is room for one or two questions. I would just wanted to ask you what you have seen until now in the patients and what has been the investigator's view.
Mm-hmm. Yeah, I mean, our studies are single-blinded because no surgeon can see, she have to see, because she is asked to take biopsies from the tissue that lights up, so she can see it, and she can see actually very nicely that the cancer lights up, and that is really positive. So on the one side we know that it will light up the cancer, and on the other side we know that it's well tolerated. So the next step is to understand how much benefit can we actually provide, how good is it, and that will come in steps up to the histology as we get later on. But we think it demonstrated that it works.
Okay. You're using UPAR as the target in order to localize your drug to the cancer tissue. Why is it a need to increase the dose? Does the drug disappear in certain other parts of the body so it doesn't find its way to the tumor, or what's the reason for going after different doses here?
Mm-hmm. Oh, that's a good question. I mean, we have to, when we select the dose, when we go into the pivotal phase, I mean, the regulatory agencies will ask us that we have picked the right dose. Also, investors will ask us that we have picked the right dose, and we, of course, would like to pick the right dose. So we have to generate the data that can document that we have selected the right dose. And what we see is that we already saw light on a very first dose. In one of the 3 patients, we saw light. I mean, when I say light, I mean contrast. We saw that the cancer light up compared to the background. But what we see when we increase the dose is that the cancer lights up more and more. And now, and that's why we are certain that we are close to selecting the optimal dose, is that the cancer get saturated. So by higher dose, we do not get more product into the cancer. So what we see with the latest doses is that the background as well is coming up. So the background lights up more and more, and the contrast disappear. So we know now that we are on a limit where we have the cancer is lighting up the maximum, and that we just have to reduce the background a little so we get the optimal light for selecting it. But we saw light in all the dose group as we gone on, but we would like to we are dosing also, I must say, we're dosing not for the weight, but we are dosing per patient. And we also see the homogeneously result as we go up in dose, so less depending on patient weight, the patient situation. So we get more robust data, and we get, more contrast as we go up. But we are hitting the ceiling, so to speak, now, because we get the background up, and that's where we should stop.
This saturation, the information from that, is that something that you can use when you go after other types of cancer, or each cancer very specific when it comes to this UPAR and how to dose?
I mean, the UPAR side of it is very similar between the cancer indication, but there's one thing that is important. In the brain, we have this blood-brain barrier. We don't have that for other cancers. So we are actually discussing if we should have 1 or 2 doses in our next indication, but we know quite well what kind of dose level that will be optimal for the rest of the cancers. And in this study, we had a non-brain derived cancer, and we actually saw very, very nice light there as well. So we have a good sense for what the dose would be also in the prevalent indications as we go on. But we may have to take 2 doses to start with to be completely certain.
Okay, great. thank you very much, Morten, for a very interesting